Well, welcome everyone to today's webinar. My name is Dawn Van Dam, and I'm the president of 1 Million Solutions in Health. Today our webinar is about accelerating drug discovery computationally, improving the efficiency of compound optimization using active machine learning. So I'm going to first give you a little bit of background about 1 Million Solutions in Health and talk to you about a special offer that we have today in regards to this technology, and then move as quickly as I can to get into the meat of the presentation so you can learn about our speakers today. So that's our panelists. Oops, I'm just going to go back for a second here. Here we go. So today I'm going to tell you about an opportunity to join our Technology Evaluation Consortium. We have put together a collaborative partnership within the pharmaceutical and biotech industry and other stakeholders. And we've had this for about 10 years now. And we're very excited to be launching it and expanding it into the marketplace and giving you the opportunity to participate in this program. So in regards to our Technology Evaluation Consortium, we all know that it's really important to collaborate in the pharmaceutical and biotech industry because things are becoming increasingly more expensive, increasingly more challenging, and it's important for people to get the efficiencies that they can by collaboration. And so actually about 10 years ago, the pharmaceutical industry approached us and asked us to put this consortium together in order to help them become more efficient at the work that they're doing. So as it says, oops, sorry, I went ahead of myself on my slides there. So the goal of this consortium has been for us to help streamline the evaluation of new technologies that support the drug development process. And to date, we've actually conducted 30 projects, and we've had quite a bit of success across the board. Here's just a partial list of those technology providers that we've brought to the pharmaceutical industry, companies like Pfizer, companies like our presenters today from Sanofi and Lundbeck, companies like AbbVie and Janssen, division of J&J. So we have some of the major organizations in the industry, and we have a real chance to help them understand and accelerate and accelerate the adoption of these new technologies. So back in June, we announced the expansion and globalization of the Technology Evaluation Consortium. And really the goal there was to say we want to further build collaborations around data sharing and meaningful technology integration. So we know that collaborations are key, and we're really building a community through 1 Million Solutions in Health to share these ideas. Today we're going to be talking to you about the third bullet there, a Technology Evaluation Consortium project, and the opportunity for you to join this project. But also we offer exclusive webinars. For instance, we're putting together a partnership with the NIH to bring you webinars that can't be accessed anywhere else. We also have our Signature Square program where we give you a chance to evaluate new technologies on a more high-level basis. And we're also putting together a technology curation database which will help you understand and look at both historical information and up-to-the-moment information about what's happening in the technologies that all support the drug discovery and development process, as well as new chemical entities. So as I said, we've been around for about 10 years, and we have a great structure to help bring forward this technology and have it be evaluated in a scientifically accurate and collaborative manner. The scientific part of all of this is headed up by Dr. Ernie Bush. He's our scientific director and has spent over 30 years in the drug safety and development area, so he brings a lot of expertise and strength to this organization. As you probably all are aware of this book, it's called Crossing the Chasm. And really the point is you don't want to fall, you have some great technology. You don't want it to fall into that abyss, that chasm of going nowhere. You don't want to run out of money before you're successful. And so part of our passion at 1 Million Solutions in Health through the Technology Evaluation Consortium is to help get people over that chasm and into a phase where they can be evaluated and understood by the end user, and therefore can find their right place in the market and be more successful with the industry. We conducted a study last year where we actually asked industry participants to tell us about the decision-making process that they go through in evaluating new technologies. And it was really quite interesting what we found in that study. What we found overall is that when we asked the question, what were your key concerns in adopting that technology for the first time for your organization, we asked them to think about the last thing that their company had adopted from a new technology perspective. And the number one thing that they said was to have validation of that technology. And that's what the Technology Evaluation Consortium does, is it helps validate the technology, helps people understand it, helps people tweak it, helps people figure out where does it fit in this drug discovery and development process, or perhaps even something that we want to bring on board, we want to absorb into our own company, and we want to be successful with ourselves. And when we asked them too, what were you evaluating, the breadth of what people were evaluating was huge. There was people looking at antibodies, biomarkers, chemistry solutions, cell systems, data collection and analysis tools, drug delivery tools, equipment, high content, high throughput tools, imaging, omics, model studies, as well as preclinical safety, sequencing, genomics, RNA, and DNA. So these are all the areas where we're getting involved in, and today we're going to bring you one of those analytical tools to look at. So our goal is to talk to you at the end of this presentation about growing our biopharma and government membership in the Technology Evaluation Consortium. And we are offering anyone listening to today's webinar an opportunity to be involved in one project for yourself, and we're hoping that you choose quantitative medicine. And what happens is you give us a brief application and the steering committee approves you, and then we're off to the races. So with no further ado, what I'm going to do right now is I'm going to turn back to the presentation for today, and I'm going to give you a brief overview of the presentation and our speakers, and then we'll jump on from there. Oh, one thing I forgot to do before is just to tell you a little bit about us, and our goals are to help improve healthcare one solution at a time at 1 Million Solutions in Health. And really the opportunity here is to improve healthcare, accelerate life sciences research, and share patient and consumer-focused ideas and solutions. So whether you're in academia, government, entrepreneurs, or businesses, please connect with us so we can share your ideas through things like today's event, a live event, a discussion forum, an article, etc. So let me take you to today's live webinar and tell you a little bit more about it, and then let our speakers take over for today. So in developing drugs, it is important to optimize for a specific set of characteristics. Many of these characteristics can be measured experimentally after purchasing or synthesizing compounds. The high cost and time required to synthesize novel compounds makes it very important to rapidly identify optimal structures. Quantitative Medicine's Computational Research Engine, or CORE, is designed to optimize. It utilizes sophisticated machine learning techniques to direct synthesis efforts to efficiently identify optimal structures. In this webinar, you'll have an opportunity to participate and ask questions about the CORE technology as we review some of these previous studies. And then, as I mentioned, additionally, we'll be offering an opportunity to further evaluate the CORE technology through a free trial to join the Technology Evaluation Consortium and participate in a project. So as we're going along, if you do have any questions, just put them in the chat box and send them to our host at 1 Million Solutions in Health, and then I will say the questions at the end to our panelists, and then they will answer your questions. So let me give you just a brief background on each of our speakers for today. Jorrit Hornberg holds a master's in molecular cell biology from VU University in Amsterdam and a PhD in systems biology from the same institute. His academic research focused on control of complex oncogenic signal transduction pathways. Jorrit has drug discovery expertise ranging from target validation and molecular pharmacology to toxicology, and has worked on multiple disease indications in immunology, oncology, and neuroscience. After working at Schering-Plough in the Netherlands and at Bayer in Germany, Jorrit joined Lundbeck in December in 2009, where he is currently head of in vitro studies in exploratory toxicology. Richard Brennan. Richard is a member of the Sanofi Project Standards and Innovation Group, acting as a scientific advisor focusing on systems biology applications in toxicology. Prior to joining Sanofi, Richard was with GeneGo, now a Thomson Reuters company, where he led the MetaTox consortium that developed ToxHunter, a systems toxicology database and data analysis tool. Richard was previously Director of Molecular and Investigative Toxicology at Iconix Biosciences, where he helped develop both Drug Matrix and ToxFX, now managed by the NIEHS. Richard also has prior hands-on experience in drug discovery and development at OSI Pharmaceuticals and Cornerstone Pharmaceuticals. Richard received a PhD from the University of Warwick in the UK and did postdoctoral work at the University of Manchester and the Harvard University School of Public Health. And our final speaker today is Josh Kangas. Josh attended Truman State University in Kirkville, Missouri, where he studied computer science and did research on the genetics of corn, as well as field work studying mud turtles. He earned his PhD under Robert Murphy at the Ray and Stephanie Lane Center for Computational Biology at Carnegie Mellon University. During his time, his work was focused on developing and testing active machine learning methods to direct high-content screening campaigns. He co-founded Quantitative Medicine with Scott Bodine and Robert Murphy to allow him to commercialize and extend some of his PhD work. Josh is currently the Chief Science Officer at Quantitative Medicine. So, with no further ado, I'm going to turn the presentation over to Josh and let him begin to tell you about this great technology, and then have the other speakers today from Sanofi and Lundbeck take you through their actual experiences with core technology. Over to you, Josh.
Thank you, Don. And I wanted to also take an opportunity to thank the attendees and the panelists for joining this discussion. So what I'd like to go over here briefly would be an introduction to quantitative medicine. I'd like to discuss compound optimization as it's currently practiced in terms of the time and cost. Then also look at the current industry approach and limitations, and then how quantitative medicine tries to solve that problem. And then kind of summarize the value, and then we will move on to the two specific case studies. So Quantitative Medicine is an early-stage computational biology company, and we developed an extremely efficient, cost-effective analytic solution for optimizing compound assay selection. We call that the Computational Research Engine, or CORE. And basically, it's two important facets. It can develop very accurate predictive models, and the way we do this is by incorporating as much data as we can into the process through what we call the knowledge base. And then it also will actually direct experimentation so that you can generate effectively the data that's used to build the models, and that results in very quickly building highly accurate predictive models. And this can substantially improve pharma productivity. Okay, so the compound optimization is one area where our technology can be applied, and right now it's where we're primarily focused. The current industry approach to drug discovery is that you want to run experiments that you expect to yield a desirable outcome. And according to Paul et al.'s Nature Drug Discovery paper from 2010, basically the results of this type of effort is you have a 96% attrition rate, takes more than 10 years to develop a single drug, and it costs more than $1.4 billion per release drug. The core approach is significantly different from this in optimization, in that we actually want to run experiments to learn how to find the desired outcomes. And the result across many studies we've done has been anywhere from 40 to 90% reduction in experimentation. Now, if you take, say, 50% reduction in time and optimization and apply that to the cost model in the Paul paper, you'd find about a $243 million savings, capitalized cost savings. So let me talk a bit about the current approach. So think of this grid as a grid of experimental results. So the rows are a set of assays that can be run, and the columns represent compounds that can be tested in those assays. So each unit in this grid then represents the result of testing a compound in an assay. So I put this green box here to indicate what we call the experimental space of interest. So this is a subset of assays and a subset of compounds from which researchers can choose which experiments to run. So again, the standard approach is to run experiments expected to yield the desired outcomes. And the way you determine which experiments is you build a predictive model from whatever data you have available from running those experimental assays historically. And so representing data here with the red pixels. So this is often done in a cycle. So you have a set of compounds, set of experimental assays, you combine the results, and then using those results, you build some kind of predictive model that allows you to predict what we're calling the drug utility for a compound. What I mean by drug utility is how well does a compound match a desirable profile for a drug. So if you have low drug utility prediction, you're basically going to ignore that compound. If you have a very high drug utility prediction, you might move that compound on to the next phase. If it's just high or a medium utility prediction, you might run that back through the assays and confirm some of those predictions. So now I have a slightly different view of the same cycle. So on the left here, we have the standard R&D cycle. On the right, we have the core R&D cycle. So the primary steps in each of these cycles are identical. So you run some experiments, and then based on the results of those experiments, you generate a predictive model, and then from that predictive model, you select a set of experiments, and then you continue until the project ceases. So in the standard approach, the difference between the standard approach and the core approach is that you use just assay data from your assays of interest, typically in the standard approach. In the core approach, you use the knowledge base, which is an enormous amount of data which comes in. I'll talk a bit more about that in a few seconds. Then when you're looking at the experiment selection, the difference between the standard approach and our core approach is that with standard approach, you choose compounds which have desirable characteristics based on your predictions. And with core, you choose the compounds which are most informative. And so kind of a nice analogy for this is thinking about the game 20 questions. So quick overview on 20 questions. Basically, the idea is one of the players thinks of an object, and the other player asks a set of yes-no questions to try to guess what that object is in less than 20 questions. So with the standard approach, what is often done is effectively you ask enough questions such that you can determine that that object is, say, a mammal, for example. But then once it's determined that it's a mammal, then you start guessing, oh, is it a fox? Is it a lion, a rabbit, whatever? And you end up spending more time without learning a whole lot more information. And sometimes you'll find the answer quickly, and sometimes you don't. Core approach is that we actually want to, with every round of this process, continuously learn to improve the predictive model. So for in the mammal example, we might then ask, oh, is it a carnivore, is it a herbivore, is it aquatic, that sort of thing. So at the end of the day, it's all about consistently trying to learn as the process continues. Okay, and then a little bit about the computational research engine and the way it operates. So basically, it's a software service that works iteratively, and we can work on any small molecule data. So these data are transferred back and forth between core and the researchers in the form of spreadsheets. So we use small molecule data that's either discrete or continuous. We don't need any units of measurement for the data, and the data can come from any source. So across your enterprise, past campaigns, any partner institutions, academia, consortia, et cetera. And because of the way the knowledge base is designed, it's really applicable to any therapeutic area because we have so much information from so many different sources, we can often find solutions to difficult problems very quickly. And this is a service which is offered in the cloud. And the effect of that is that we could scale very rapidly. And so that means we have unlimited concurrent studies. It also means that when data is submitted to our system, we can very quickly turn around results. And the result of that is saving you time in deciding which compounds to run next. And also, because we're using these cloud results, we can keep your data highly secure. So as the cloud instances are running through your work, once they're done and they've returned the results to researchers, then these instances destroy themselves and there's no permanent storage of your data anywhere out in the cloud. So it's kept very safe. Okay, so coming back to the knowledge base, if we look at the experimental space we had defined before, now I've added in some more data to the experimental space, and what we find is we have about 200 million results, and it's less than 1% coverage, but we use all of that data to make accurate predictions in the little green box. All right, so the core knowledge base right now is a database of public screening data. We have about 200 million results, as I was saying, and it comes from various sources, sort of high throughput screening, high content screening, in vivo tests, clinical trials, you name it, we probably have results for it. And again, it covers many therapeutic areas, and we're trying to take advantage of all this knowledge from small molecule research and pull it together from many unrelated sources to make really accurate predictions. Because we have this flexibility, we can also pull data in from different phases of the research process. So in this particular example, we have different campaigns, say, in one institution, and we can use knowledge that we learned in the lead optimization for campaign 3 to actually have an impact in the hit to lead phase for campaign X. And the idea here is that we want to get useful information to a project as quickly as possible so that we can dramatically reduce later stage attrition. So we come back to this knowledge base, and what ends up happening is that you can add in your own your proprietary experimental data temporarily to the knowledge base and then run the same process. And then when you've completed that, then your data is gone. Now, one of the potential issues that can happen when you incorporate so much data like this is that you may find that there's a lot of noise in the data. So that's absolutely the case in public data sets, that you find a ton of noise. And Core has a unique way of handling that. So on the one hand, Core uses machine learning methods that can tolerate the noise when making predictions, so they're rather robust to noise. But beyond that, Core has the capacity to actually direct experimentation. So while it's directing experimentation, it recommends that experiments are run which allow you to test your correlations that you find. And if correlations are spurious, as you add more data to the system, per the recommendation of the Core, those spurious correlations will basically wash out, and the good correlations will come to the top and be used for making predictions in your experimental space. Okay, so coming now back to the cycle that I was showing before, I want to point out the major differences in the ways that currently experimentation is directed and the way Core directs experimentation. So the most important part here is accurately predicting drug utility. And so looking at Core, we also want to focus on predicting drug utility. So the two differences, though, that one, we use this knowledge base, and so the knowledge base gives us a lot more information that we can use to make accurate drug utility predictions. The other part is that with every cycle of this process, we try to assess what we're calling information utility, which is how much can be learned from testing a compound. So even if a compound does not look like it will be a good drug, it may give us enough information that we can make better predictions for drug utility later on. And we can also, as we're going through this process, assess how much information could be gathered in total from all of the compounds that we're considering. And if we run this process a number of times, that allows us to measure the progress of a campaign. So what we've called that here is a stopping rule. So this is a way in which we can assess a project as it's going along. one reason you might stop a project is because you have found a good compound and it meets all of your criteria and you might move along. But on the other hand, you want to be able to estimate whether or not you found the best compound in your library. And if you found the best compound, then you have a couple of choices to make. Either you go ahead and move forward with that compound, or the best compound you found is not good enough, and you might need to add more compounds or add more assays or basically reassess your project. And so what happens here is because you have this control over the way experimentation is done, you can see some really dramatic reductions of anywhere from 40 to 90% in your experimentation. And so long term, you have the ability to build really informative data sets for your future campaigns because CORE is directing the experimentation to find the most informative compounds. And then you're continually learning more accurate predictive models that are used throughout the process. And then because you can use the models and the information learned from many different phases at the same time, you can reduce late phase attrition. And so again, coming back to our estimate from the Paul paper I mentioned early on, that could be around $243 million in capitalized cost savings with a reduction in time and cost by 50% for optimization. So our research over the last 5 years, as well as about 20 studies with industry partners, has really strongly supported these sorts of cost reductions. So without further ado, I would like to turn it over to Jörg Hornberg from Lundbeck to discuss hepatotoxicity and an optimization study.
All right, thank you, Josh, and Dawn also for the introduction. What I'll do today is give a brief overview in headlines of our exploratory toxicology strategy at Lundbeck, and then also talk a bit about the pilot projects that we have run with quantitative medicine through the Technology Evaluation Consortium. It starts with obviously an overview of how big a problem drug safety actually is. Most of you are probably familiar with this, but I just wanted to point it out again. Toxicology and safety is really a major reason for attrition in clinical development. It is perhaps a little bit increasing even if you look at the recent literature. Besides that, also when drugs are withdrawn from the market, it is usually due to human safety concerns. And also there's often actions from the authorities in terms of warnings that are added to labels or use restrictions that are imposed. And this is obviously a problem for the pharmaceutical industry. When we ask ourselves the question, how can we actually improve this? Of course, safety and toxicology studies have traditionally been carried out in preclinical development, basically to assess whether a drug is or drug candidate is safe enough to dose in humans and at what dose. But over the past probably around 10 years, more and more predictive safety and tox studies have been implemented, also in earlier phases of the drug discovery and development value chain, with the idea to basically if you can fail early, you will fail cheap. So if a compound is not going to make it because of safety reasons, you better find out early. So sort of safety and toxicology testing has gone beyond sort of traditional methods just to assess whether a compound has been is safe enough to dose in humans, but also look at what might happen further down the line if we expose a large patient population. Of course, if you think about it, failing early is not necessarily the same as increasing the success of drug discovery and drug development, because if you deselect compounds relatively early that have a potential safety issue, that does not automatically imply that you will also identify or increase the identification of safe compounds. So when we thought about this, we basically came up with the reasoning that exploratory toxicology must be an integrated part of the drug discovery process. You think about it, the development candidate is a fixed molecule that has all the properties, including those that cause toxicity, fixed, whether it's related to the target it hits, secondary pharmacology, or the chemistry. And in order to take those things out of the molecule, you have to actually design safety into the molecular entity before the development candidate stage. Basically just like any other drug property. So the idea here is that to identify potential liabilities early on in the process, this is the failing early part, but also to provide a way forward in order to mitigate those potential liabilities. So provide directions to the drug discovery teams and thereby increase the likelihood that a drug will not fail. So if you just look at the various phases of drug discovery, obviously this starts all with target validation, and as part of this process, the first thing we do is do a thorough safety assessment of the novel drug targets that are proposed. And very briefly, this takes into account all knowledge we can basically find to generate an overview of potential safety issues that are related, that you could expect when you hit this target, or perhaps when you look at other pathway components, or by affecting the particular cell type that you are trying to affect. It could also involve adverse effects that are known of drugs that have, for instance, a comparable mechanism of action. And very often this leads to follow-up experiments. This could be dedicated target safety studies. It could also be that safety endpoints are included in target validation studies, or the specific safety assays or secondary pharmacology assays are implemented in a project screening tree or flowchart through which the compounds flow in a project. And ultimately then you get an integrated target safety assessment that obviously is placed into context of the project. So what is the indication and the medical need of that indication, what is the competitive environment, etc. And then the next question is when should you actually start testing compounds? And this could be a matter of debate. Some say, well, during the lead optimization phase, compounds change so much, so the data that you generate for very early compounds are not representative for the final candidate, so don't start testing too early. And my take on that is that actually goes for all properties that you assess early, including target potency, including stability, exposure, and other things that you look at in that phase. So what we actually do is we already in the hit to lead phase, where basically all hits that have been found in screening campaigns, for instance, are being assessed and looked at from different angles. In terms of course, a lot of potential properties that are relevant for a chemical series, not just potency, but a lot of things, including IP perspective, lipophilicity, selectivity, and also for a company like Lundbeck, focused on CNS disorders, does this compound series, is it possible to actually enter the brain, and a lot of things. And basically we have made toxicology part of this, and the idea here is not to already at this early stage provide go, no-go decisions, but just to flag potential safety issues that might be inherent to the chemical series, and thereby have an impact on the prioritization of series that will be taken forward then into lead optimization. So the idea here is that you identify chemical series without inherent safety issues. And then during lead optimization, compounds are going to change a lot. There's a lot of analogs being made, and our aim in this phase is to identify compound-related risk factors and also design them out. And so the idea is that you assess how structural changes that you have in this phase affect certain safety endpoints, but also if you have identified potential liabilities, to actually determine structure-activity relationships and thereby optimize the compound series on the safety endpoint. And then towards the end, when the development candidate is selected, we obviously do a very broad toxicological profiling of the potential candidates, and this includes things like to follow up on this target safety assessment if it has not been followed up on yet, but also to get a good insight into the acute in vivo toxicology and safety pharmacology issues that there might be, trying to establish a therapeutic index, and also, of course, generate a plan, a translational plan towards dose range finding studies, the GLP tox studies, and into the clinic. So what is very important in this strategy is that you apply the right technology and the right tools. And Don, as you already pointed out, one thing that is very often mentioned as a hurdle in terms of implementing new technologies is the validation. And when people speak about validation, I think in the safety area, what they mean is the productivity of those assays or technologies towards clinical safety, so that you can use the new technology or method to predict whether a compound is going to be safe for humans or not. And I think that is something that we sometimes do in-house, sometimes technology providers do it themselves, but also often we do this in collaboration with others, such as through the Technology Evaluation Consortium. I think this is a very good way of interacting with technology providers simultaneously as with other pharma companies, and thereby sort of together discuss pre-competitively how novel technologies could actually impact the predictions of compounds being safe for humans or not. But besides this productivity, of course, they also need to have a certain throughput, especially if you consider the early phases, such as the hit-to-lead process, and in particular the lead optimization process, because you need to screen a lot of compounds. So this is usually in vitro. You don't have a lot of compound available. You want the data quickly, and also the data need to be quantitative in a certain way so that you can use them for decision making. It's often not good enough to say, well, this compound is toxic or moderately toxic, but rather you'd like to provide a % inhibition or an IC50 value, something that a chemist can work with in their optimization. Just as an example, this is a high content screening assay that we've developed that we can use to catch about 50% of idiosyncratic liver toxicants, and that we actually actively use in our drug discovery programs. So now that we want to optimize on safety, I think very often in projects we find that this is actually possible. There's a lot of these assays now out there. There's a lot of technologies that are still emerging, but also we find that often it is very difficult. Drug discovery is difficult enough as it is. You're already optimizing on a lot of things, and then to also include on a number of safety readouts, and they're often quite many, that of course places a huge demand on the medicinal chemistry resources. And this is in particular difficult if there is no apparent structure-activity relationship or apparent to the chemical eye. So in those cases, I think predictive QSAR modeling can help out. And just as an example from our own efforts that we do in-house is there on the bottom. There's two examples, one on cytotoxicity and one on hERG. So for instance, the one on cytotoxicity on the left column, you have compounds that were predicted to be cytotoxic, and of those you can see in red the ones that were actually then measured to be cytotoxic. The green ones were then clean, even though they were predicted to be cytotoxic. And then the second column is those compounds that would be predicted to be not cytotoxic, and as you can see, most of those would indeed not be cytotoxic. And then there's a column on where the model is not sure, and a column on where they are sort of the compounds are out of the domain of the model. And then for hERG, you basically can see a similar distribution. So when we got the opportunity to work with quantitative medicine through the Technology Evaluation Consortium. We already had some experience with quantitative SAR modeling and using that in a predictive way to advance our projects. But of course, as Josh also presented, there's some aspects of their technology that we thought was very interesting, so we decided to run a couple of pilot projects. And the first project is where we provided quantitative medicine with data on 10 in vitro safety readouts on about 100 drugs. Those were drugs from the public domain with a known clinical hepatotoxicity profiles. Structures were also known, and obviously quantitative medicine has their own database. And what we did there was try to predict which of those compounds were hepatotoxic and which not. So the data set was randomly divided into a training set, 80% or 80 compounds, and a test set of 20 compounds. And each round, one compound was selected by quantitative medicine, for which we then revealed the data. And each round, then the predictive model was updated or constructed, and then all compounds were predicted. And what you can see here on the is basically the result. So on the y-axis, you have the area under the receiver operating curve, basically meaning if that would be 1, you'd have a perfect prediction. If that would be 0.5, it would be sort of a random. You could just as well flip a coin, basically. And then we have a red line, which is random forest modeling, and then the core technology from quantitative medicine in blue. And as you can clearly see, the core technology goes into a predictive range of about over 75%, which is quite high compared to if you, for instance, just compare to the experimental. If you would only use the experiments, you don't reach that predictivity. So then I think we maximally get to about 2/3. So this was a good improvement on that. And also you can see that the core technology also keeps learning as more compounds from the training set are basically being tested. So we thought that was very interesting. Of course, these were also compounds that are in the public domain, and we did not exactly know how many of these compounds were part of the database quantitative medicine has. So we decided to run a second pilot study where we actually gave them compounds from a closed project at Lundbeck, including all the structures, and also data on 3 assays, the primary screening assay on the target, but also 2 in vitro tox assays. And there were about 185 compounds with data on all 3 readouts, but I think in total, for instance, on the primary assay, had about 600 compounds with data on that. So they got the structures. And then we also provided the data, the actual data for the first 40 compounds that we had synthesized in the project. And then what they were asked to do is try to, as quickly as possible, identify the compound with the optimal profile. Optimal profile meaning the best potency on target that you can possibly get within this compound set, but also they should be clean on the 2 in vitro tox assays. And what they've done is at each round of simulation, they selected 10 compounds from the total compound set, for which we then revealed the data. Basically every time then in each round the predictive model was updated, as Josh explained. And we used we compared a few ways of how to select compounds. One was greedy selection, which is basically choosing the compounds that are predicted to be the best, to match closely this desired profile. Also the core selection, which was more focused on the one hand, finding compounds with the optimal profile, but also finding compounds that would provide most information to the model, as Josh already explained. And then, of course, we had the actual campaign as how it went in reality. And what you could see was that with core technology, the best compound was identified after 7 rounds, whereas in reality, we had identified it after 10 rounds. And a second thing that we in this project also assessed, and I think this is actually very important, is when in a project should you actually stop making compounds. And sometimes you're not aware that you've actually already made the best compound that is possible within the chemical space that you are exploring, and projects continue. And knowing that you've actually already found the best compound possible might actually also be very nice. And maybe this is something that Josh can explain later, but we discussed how we should set up this stopping rule, and basically it tells us where the model basically does not become more, doesn't gain more information, and also not more better compounds are being found. And in the core selection method, the best compound was found after round 7, as I said in the previous slide, but also after 8 rounds, they already told us to stop testing, basically. Whereas if in the actual campaign, we actually went on 6 more rounds after we had found the best compound. But just to conclude, the outcomes of these studies that we've done with quantitative medicine is that prediction of hepatotoxicity improved versus only doing experiments. I think that's, in general, a good thing to realize, is that modeling does help, in addition to experiments, to improve predictions. The search for the best compound was accelerated with around 30% in the particular case. And also, we clearly see that we could get additional productivity from core compared to, for instance, random forest, also using greedy selection. And then the stopping rule, that I think that is also an interesting tool to estimate whether you can make progress in the future or not. And then just a general comment that I think predictive QSAR modeling is an interesting tool to aid in guiding optimization projects. One thing that Josh mentioned was prioritization of experiments, but also I think when you have an ongoing program and there's a number of, basically a very large number of potential compounds that you could make and then test, I think you can use this also to prioritize which compounds you should make. And there will be different compounds, especially if you use the core technology, because it will not only be focused on finding the best compounds, which is usually what we try to do, but also try to gain more information. And obviously, I think it's also very valuable to determine whether the chemical space has been exhausted. I think that was very interesting. So with that, I'd like to just conclude and pass the ball on to Richard from Sanofi.
Great. Thanks, Jorrit. I'd like to just quickly take the opportunity to thank Josh and Dawn for the opportunity to speak today, and Dawn for the introduction. Also, I'd like to quickly thank Jorrit, actually, for his very nice introduction to the rationale and process for early screening in predictive toxicology, and the need for truly predictive and useful assays, fit for purpose assays. And what I'd like to talk about today is a pilot project operated through the Technology Evaluation Consortium with Quantitative Medicine to look at whether we could leverage a very large public project through the EPA called ToxCast, which I'll talk a little bit about, which has generated a lot of data and which we actually collaborated with. Whether we can leverage the data generated through this process to identify potential new screening strategies for predicting preclinical toxicities or safety concerns that typically would stop or would cause concern in a drug discovery program. I'm going to start off just with a few acknowledgements. So obviously this isn't just me working this project. There's a lot of people involved, and particularly here at Sanofi, Doug Keller started off this process. He's the head of the preclinical safety part of Project Standards and Innovation, which is the group I work in. We have also got people from Frankfurt. Friedman Schmidt is in our computational toxicology group, and Rich Karl-Malik from our Bridgewater group was involved in a lot of the statistical analysis of the outcomes. And quantitative medicine, Josh Kangas was our direct contact there. Very good people to work with, very responsive, and a good understanding of what we were trying to do. And again, at the Technology Evaluation Consortium, Dawn Van Dam, who was a great project manager for the whole thing, bringing everything together, working with contracts, setting up meetings, those kinds of things, and Ernie Bush, who was intermediary in data exchange and acted as an independent conduit, if you like, for exchanging training sets and validation sets of data. Just a quick word about the Technology Evaluation Consortium, which is now operating through 1 Million Solutions in Health. When we joined the consortium, it was part of Cambridge Health Tech Associates, and as Dawn mentioned, it's a mechanism for bringing companies together to do a coordinated evaluation of new technologies and assess their applicability to, in our case, thinking about safety assessment uses. We chose this particular project both to help us understand whether there was any value in the ToxCast data set, which is a very large data set, large and complex data set, but also to take a look at the quantitative medicine approach to machine learning and active learning to evaluate the potential uses of core technology in other programs as well. A quick word about the ToxCast program through the EPA. For those of you who may not be familiar with it, this was one of the earliest responses to a report from the National Academy of Sciences on how we should really be approaching safety assessment in the 21st century and moving away from, as much as possible, from expensive in vivo animal experiments to trying to take a more mechanistic and target-based approach and try to do things with mechanistically based in vitro assays. ToxCast program is independent from the Tox21 consortium, but it's very much aligned with that. And again, the Tox21 consortium is pushing towards this concept of in vitro screening for predicting toxicity outcomes. So at a very high level, what ToxCast has done is it's taken a large number of chemicals for many of which there's quite an existing body of in vivo outcome data, so toxicology data, pharmacology data, long-term exposure data, population data in some cases. And they've taken those chemicals and run them across a very large battery of in vitro assays. I'll talk a little bit more about that in a second. A wide range of assays covering a lot of different endpoints. And they've brought all those things together, including some PK/PD modeling to estimate exposure, to look at computer simulations that try and tie together the in vitro assay data to the in vivo outcomes and come up with ways of identifying predictive modeling for in vivo outcomes for these chemicals. If you want to find some more information on that, it's available at the EPA website. You can just do a quick Google for ToxCast, and that should take you pretty much right there. The data set we're working with for this study is part of phase 2 of ToxCast. We got involved in phase 2, and as part of phase 2, the EPA screened more than 2,000 chemicals across 700 in vitro assays, so really broad swath, almost shotgun screening of available in vitro assays. These are biochemical-based assays, they're cell-based assays. They measure target activity data. They also measure cell response data and pharmacology data. For many of those compounds, as I said, there's also a lot of in vivo data available. And there's a lot of overlap between the in vivo data, which EPA has put together in a database called ToxRefDB, and the in vitro data. So you can try and think about doing this correlation between in vitro and in vivo outcomes. As part of phase 2, several pharma companies got involved in this exercise in ToxCast, and we donated between 135 compounds between the 6 pharmaceutical companies involved, and 37 of those compounds came from Sanofi. All of those compounds were discontinued in development for one or more reasons, some of which were for safety outcomes, and some of which were for other reasons. But for all of those compounds, we also gave EPA access to all of the available preclinical study reports. So all the in vivo animal experiments relating to those compounds were available and were incorporated as far as possible into the ToxRef-DB database, along with the compound information on the compound structure, the compound target, the indication, and all those other useful types of data. So as well as the pharma-donated compounds, there were several hundred other compounds in that 2,000 chemical database that have some pharmaceutical relevance. The objective here was to really systematically mine the data generated by the in vitro assays on these compounds and see if we can predict toxicity endpoints of relevance to our preclinical safety assessment studies. In order to do this, we did a pretty intensive mining of the ToxRef-DB database to identify compounds that had particular phenotypes that cause particular issues in animals. I'll tell you a little bit more about that in a second. And then we used the quantitative medicine approach to do machine learning to try and identify from that assay data, from those 700 in vitro assays across 2,000 compounds, whether any of those assays or combinations of those assays were able to predict the outcomes that we were concerned about. So I won't talk in detail about the core process, but Josh has already described that quite nicely. But really, from our point of view, what we wanted to do, as I mentioned, is ask three really key questions. Is there any predictive value in the ToxCast screening battery for endpoints of concern to preclinical safety in a pharmaceutical R&D paradigm? Can the active learning process, can the core technology help us improve the efficiency of assay identification, feature selection? And is it possible that the database behind core can also add to that predictivity by identifying other assays that are in the database but not necessarily as part of the ToxCast screening battery that may also have predictive power? In terms of endpoints, we picked some endpoints that are really things that when we run an animal assay in a preclinical safety assessment, are things that would typically cause us concern. They're also chosen on the basis of availability of compounds in the data set in the ToxRef-DB. Are there enough compounds that represent these endpoints that we can build a robust enough training set to really identify predictive assays? So we did a systematic mining of ToxRef-DB. We identified compounds that had one or more of these endpoints. It was also important to identify compounds that did not cause those endpoints. So you need positive compounds and negative compounds for your training set. And having identified all those compounds, we did a random split of the data set to remove about a quarter of the compounds from the data set to use as a forward validation exercise. And we provided the remaining 75% of the compounds and the associated ToxCast in vitro data to quantitative medicine to do the machine learning model building exercise. In terms of training set size, the number of compounds varied between the endpoints. Some endpoints had fewer positive compounds than others. So there's quite a nice distribution across the endpoints of interest of small positive versus larger negative training sets to about balanced training sets to actually over-balanced in terms of positive compounds and negative compounds. But in general, for each of these endpoints, we thought there was enough compounds and enough data to generate and validate whether there was any predictive data in the ToxCast data set. For data analysis, once quantitative medicine had done the model building process, they provided us with the best model for each of those endpoints. What we did was then send them the validation compounds and the data, the in vitro data for those validation compounds, without telling them the true phenotype, the true outcome with those compounds for the endpoints in question. And then they returned to us their predictions for each of those validation compounds as to whether they were positive or negative for the endpoint in question. In terms of evaluating the models, we looked at compounds where we were able to make a solid call of positive versus negative against a particular endpoint. So, for example, did or did not cause liver necrosis. And also for which the quantitative medicine model was able to make a call as to whether it was positive or negative. There were a few compounds in there where, probably because of missing data points, the model was not able to predict for a given compound. And then as Jorrit described, we use a receiver operating curve as a general, the area under the receiver operating curve as a general readout of whether or not there was any predictive power in that model for the endpoint in question. To look at the active learning and core technology, the process was that quantitative medicine applied an iterative learning process, starting with adding in small numbers of compounds to build the predictive model and assessing the accuracy as they were going on along there. But more importantly, from our point of view, looked at that across a number of different data sets. So what data the model was allowed to use to try and make a prediction. And that was looking at either just the in vitro data from the ToxCast screening battery, looking at the structure of the compounds, or fingerprints generated from the structure of the compounds to build quantitative structure-activity relationship models, similar to what Jorrit was describing previously. Having access to the entire knowledge base behind core, which includes all of the assays in that knowledge base, as well as the structural features, and then finally combining all those things together to see whether adding in core features on top of structural features on top of the ToxCast screening data gives a better model. So how does that look in terms of results? What we're looking at in this table is actually, for each of the endpoints of question, the area under the ROC curve, given different feature sets available for the modeling process. So as Jorrit described, area under the ROC curve, quite simply, if you've got an area under the ROC curve of 0.5, then really there's no predictive value in that model at all. It's like flipping a coin. It's deciding whether the compound is positive or negative. As the area under the curve increases, that model has more and more predictive power. So if we look here at the data for the ToxCast in vitro data and the models that were built in that, in no case was the in vitro data alone sufficient to generate a well-performing model, and in none of those cases did the in vitro data alone give us the best predictive model. And in fact, in several cases, there was no predictive power in that in vitro data set across all 700 assays for the endpoint that we were concerned about. This is not necessarily a surprise. What we're looking at here is trying to correlate endpoint data, in vivo data, multi-mechanistic data to particular biochemical assays. And we knew going into this that would be a particularly difficult problem, and didn't necessarily expect there to be a lot of predictive power in the assay set. This is rather different to what EPA is trying to do. They're concerned about mechanisms of particular concern, such as endocrine disruption. They're concerned about low-dose exposures over long periods of time and population effects, reproductive effects, more long-term things than we in the pharma industry are necessarily concerned about in assessing safety. So not a lot of power in the in vitro data. Again, not surprising, but a little disappointing. If we look at the other sets of features that core is able to bring to the table, we find that in some cases there's some predictive power in just the compound structures, but again, not a great deal there. In one of the endpoints, we found that compound structure alone gave us about as good prediction as you can get. When you bring in the core database and look just at the core database, actually for 3 of the 7 endpoints we looked at, the core database alone generated the best model for that particular endpoint. So it did better in isolation than either the compound structures, the ToxCast in vitro data, or even putting all those things together. And then in 4 out of the 7 endpoints, the best model was actually from combining all of those different feature sets together and allowing the machine learning to get access to as much of the available data as possible. Just a quick example of a ROC curve here. So Jorrit described this briefly, but if you have an area on the ROC curve of 0.5, the curve itself looks like a straight line like that. And what we're looking at is the balance here between the true positive fraction, that is the model's sensitivity, its ability to pick out positive compounds as positive, and the false positive fraction, and that is how many mistakes the model makes in calling compounds positive when they're actually negative. So a diagonal line is 0.5 under the curve and is basically a coin flip, positive, negative. If you've got a perfect model, the curve actually looks like that. So for you always pick out all of the true positives and never pick out any of the false positives. But actually, in practice, the curves typically look something like this with a nice distribution. And you can pick a point along that curve where there's a set of parameters that can give you a reasonable balance between the sensitivity of the assay, so true positive rate, in this case for a model that's trying to predict the compound's ability to cause liver necrosis and increase ALT. You can find that we can actually identify 77% of those compounds correctly. But the trade-off is that we also pick out 30% of negative compounds as positive at that point. So it's a balance between sensitivity, the ability to identify toxicants, and specificity, the ability to identify non-toxicants. And that's always a trade-off in predictive toxicology. And again, what the core process and quantitative medicine brought to the table was that ability to go from having a data set that's maybe not optimal, has some predictive power, but not enough to make it useful, to actually bringing other data sets into the mix and improving the performance of those assays in an iterative and efficient way. One further interesting feature of the process is the ability of the machine learning to identify which assays within the data set give you the best or contribute most to that predictive power. And this is a noisy slide, so don't look necessarily at the assays individually that are picked out here, but I just wanted to point out that for many of the assays where in the ToxCast data set there was predictive power, it's actually a very small set of those assays, 3 or 4 assays that are being consistently picked out as predictive for each of these organ damage endpoints. And these are assays that actually represent fairly high-level cell-based effects relating to inhibition of cell proliferation, so just stopping the cells dividing, or actually arresting the cells in various phases of the cell cycle. So they're quite high-level effects, and it's interesting to try and think about what that means in terms of how you should formulate an early screening battery for preclinical safety. So a few conclusions. In terms of model predictions, in vitro data from ToxCast, it really doesn't provide very much predictive power in predicting organ damage endpoints at high dose, at short-term or medium-term exposures in preclinical animals. And again, that wasn't necessarily a surprise. We didn't necessarily think that would be the case, and it really points to the best use of that ToxCast assay data in a more mechanistic understanding aspect than in a pharmaceutical early preclinical scenario. But we were able to show that the core knowledge base provides a lot of benefit. It increases the power of the modeling process, brings new data in, identifies what assays ToxCast maybe should be running to do these kinds of predictions. Really, the potential here is that for any given endpoint, then the core technology could be applied within ToxCast to identify potential additional assays to add to the battery to answer specific questions. And there are some remaining questions from this around, what does it mean that the machine learning process picked out these key 4 or 5 assays, as being predictive of those endpoints? Is there some mechanistic information in there, and maybe should we think about including that type of assay into our screening batteries? Overall, there's no predictive power in the in vitro screens for the organ damage endpoints we were concerned about, but maybe within that data set, we haven't looked yet, but maybe within that data set there are particular assays that pick out subsets of compounds with particular phenotypes of interest. So that's worth looking into. And finally, what are the features that CORE is identifying as being predictive above and beyond what's in the ToxCast data set, and what does that tell us about the mechanism behind some of these toxicants and how they're causing organ damage? And again, is there any value in any of those assays as part of a preclinical safety screening battery? So I'll finish there and be happy to answer any questions.
Great, thanks, Richard. And I think Josh has just a couple more slides, so pass the slide deck back to Josh and let him cover those off. Josh, you're not unmuted yet.
Yeah, sorry about that. So basically, just wanted to point out that your study was primarily focused on demonstrating the ability for CORE to actually direct experimentation, and Richard's study was primarily to demonstrate the quality of the predictions that can be made using CORE. And then I also just wanted to take an opportunity to say thank you to Stuart from Lundbeck and Richard from Sanofi and the people who work with them, and then John, Ernie, and Kevin from 1 Million Solutions in Health. And with that, I think questions.
Great. Well, thanks so much to everyone. This has been a fabulous presentation. We've covered a tremendous amount of ground, and of course, therefore, we have tons and tons of questions. So I will just pass those over to our panelists one by one, and sometimes it'll be obvious to me who should answer them, and sometimes it won't. But I will do my best to try to direct them at least to a first respondent. So, and just a reminder, you can still put questions in the chat box, and also we'll put up the survey in the chat box again as well. If you could answer that before you leave, that would be great. So perhaps this one, based upon when it came, is perhaps for Jort, but could you comment on the utility of high throughput screening data, for example, connectivity map or the links, in assessing the toxicity? Or do you see such signatures of use in early toxicity assessment?
Yeah, good question. I don't have experience with this particular, with connectivity maps or links yet. What we have used is toxicogenomics, for instance, where you also basically apply gene expression signatures to predict toxicity. I think in some cases it has been very useful. In some cases it has also provided mechanistic insight, and in some cases it could also provide biomarkers that you could basically use going forward. One of the things, and I must say with links, that I think that's early connectivity map. I guess it's more broader genome-wide gene expression, but I haven't seen that used much yet, and we certainly do not. But one of the things that one could consider, of course, is I don't know if this is a technology that you're particularly interested in or are involved in, but it could, of course, be interesting to discuss this as an option with Don as an option for the Technology Evaluation Consortium, because it's precisely those type of validation studies that we do in the consortium. I'm sorry, I can't answer in more detail.
Okay, great. Thanks, Jort. Is there anything that Richard or Josh want to add to that question?
I would add, just from a computational perspective, we can use any kind of data in CORE, so we can certainly give that a shot.
Okay, great, thanks. So the next question is, Today we've seen examples of core being used with great success in the lead optimization phase and preclinical phases. Can you provide an example or two of how core can be used in the hit to lead and preclinical phases? Can I expect similar time and cost savings ranging from 40 to 90% as with the lead optimization and preclinical?
Yeah, basically,
sorry, go ahead.
Any situation where you could take an iterative approach to running experimentation, you can do that. So as early as a high throughput screening approach where maybe you have plates already made up with a few hundred compounds on it or more, you can use core to select which plates to run. But then all the way to in vivo toxicity studies, if you're trying to generate data for those, we can also get in there. And I don't see any reason to expect that you wouldn't see the same sort of savings from any of those kinds of studies.
Okay, thanks. This is really a question for everyone, but I'm going to direct it to Richard. What relevance could these predictive assays have since experimental validation of a predicted effect is required anyway? And we just need you to unmute yourself, Richard.
Oh, yeah, sorry. Can you repeat the question, please?
What relevance could these predictive assays have since experimental validation of a predicted effect is required anyway?
So, yeah, absolutely. It's true that ultimately your compound is going to have to be experimentally validated. It's going to have to go through the traditional screening battery, at least currently in space. What we're trying to do here is really front load, if you like, the compounds that go into those animal studies with compounds that we believe have a better chance of success of passing through those assays. I mean, because there's obviously a big bottleneck for us, compounds failing in preclinical safety. We don't want to be doing that. By the time we get to running assays in animals, we want to be fairly confident that that compound is going to go through smoothly. So the idea is to really use these screening batteries upfront and early to prioritize compounds for further development. So Jorrit kind of described this quite well of screening out bad actors early, but also trying to engineer in safety into the molecule as it progresses through the development pipeline. So that's really the idea, is to apply these in a kind of stepwise prioritization exercise to improve the quality of the compounds as they get through higher levels of development.
Yeah, I fully agree with that, Richard. I think another aspect that I would like to add is that I think also using this, applying this to a virtual compound library, you would likely make compounds and test compounds that you would otherwise not have made and tested, because either they provide information or simply because you haven't thought about them. Whereas the models would basically direct you in a certain direction.
Great, thanks.
I would add that in initially thinking about this product, I looked at some other computational approaches to this sort of work, and what I saw is that people were really constrained by sticking to their predictive models, and they were making mistakes because they were leaning too heavily on the predictive models exclusively. And by taking this approach where we actually are wrapping the model around the experimentation process, we're not leaning on the model, we're actually leaning on the experimentation.
Great, thanks so much. So while you're speaking, Josh, a question for you. Can core work on research for biologics?
Right now, we are only working on small molecules because the knowledge base is designed to work on small molecules. We don't have the same wealth of information for biologics as we do for small molecules. But it could certainly be done in the future.
Okay, great, thanks. So this is perhaps a question for Jorrit. Do you think the quantitative medicine technology is mainly suited for applications in the safety and toxicology area?
Yes and no. I mean, mainly, obviously, they have the database, ToxCast, so they have a lot of safety data in their database, and therefore you could imagine that they perform well in the safety area. But actually, I think also after running the second pilot where we let them optimize on both safety and target potency, I think I got convinced it's more a medicinal chemistry tool, basically to find out what are the best compounds, and I think it could be used very broadly. There are obviously many modelers, QSAR modelers out there, but I think the combination of the broad data set that they have in their database, and also the way of modeling, the way of active learning is relatively unique. I think I've not seen that elsewhere, and so I really think this is more widely applicable than just in the safety area. But of course, they will perform well there in that area.
Great. Richard, here's a question for you. Given that the most informative assays for several of the endpoints were those measuring cell proliferation or cell cycle arrest, do you think it is worthwhile to include these assays in an early tox or lead optimization screening battery?
Thanks, Dawn. That's a really interesting question. I think one thing we'd need to do is to go back and look at those data to see, within those 700 assays ToxCast run, were there other cytotoxicity or cell arrest or anti-proliferation assays that had similar predictive power that maybe we just passed over because they were too similar to the ones that were picked out. To confirm that, that being said, we do know that it is quite well understood that sensitivity of cells to growth inhibition is generally higher than it is to growing cells to growth inhibition is higher than quiescent cells is to cytotoxicity. So there is an increased sensitivity of the assays in that respect. But one barrier we face there, from Sanofi's point of view anyway, in our preclinical predictive assay paradigm, is that we typically use primary hepatocytes for those types of assay. And primary hepatocytes don't divide, so it's difficult to build in a proliferation assay into that paradigm. Although I think it would be worth evaluating whether using perhaps an immortalized liver cell line and looking at proliferation, or perhaps looking at proliferation in one of our high content screening paradigms might be worthwhile. So yeah, I think it's an interesting question and something that we should look at.
Great, thanks, Richard. So this is a question for Josh. And maybe just this, great, okay. There can be a lot of noise in public data. Some of the data are generally terrible in terms of the quality of the labels. How can CORE handle that noise?
So basically, CORE handles it in two ways. One is with the machine learning methods that can really filter through noisy data to find signal. But even if a noisy data set seems to be highly correlated with whatever it is we're trying to predict, and that would be a spurious correlation, we do the recommendation of experiments to the active learning portion. Generally, those correlations are going to wash out. And if they don't wash out, it's probably worth considering why those correlations are found, and maybe there's some novel biological knowledge that can be gleaned from that.
Okay, thanks, Josh. Another question here for you is, could you comment on where the drug repositioning, actually, you could probably all comment on the first part of the question, but could you comment on where the drug repositioning pipelines stand vis-a-vis your pipelines, which seem to be more focused on novel target discovery and/or safety assessment? In other words, how can a researcher focusing on drug repositioning utilize your services? So maybe Josh can answer, and then Jorrit and Richard can comment.
Sure. So basically, the researcher would have to come up with a set of data, so compounds and their effects that would represent the efficacy in the whatever drug they are interested or system they're interested in working on. And then when they run that through core, then we can identify some compounds that will either match the profile they're looking for, or we can find correlations with other parts of the data set in the knowledge base that might lend themselves to being used in a repositioning framework.
So you gave a quick overview of how you integrate safety and toxicology into the drug discovery process at Lundbeck, but at a strategic level. How do you secure that strategy or ensure that strategy is also brought into practice in your daily work?
Yes, yeah, good question. So that is something that obviously does not happen overnight. I can name a few things. One of the things that we have identified as being very important is that you are part of the drug discovery core teams as an equal partner to the biologist, the chemist, pharmacologist, etc. And in terms of bringing the project forward, it's important to be there when people discuss novel targets, novel series, or optimization strategies to make sure that your assays are part of that. Another thing that has helped us is to really create broad acceptance of your strategy throughout the organization, from management, obviously, but also actually on the scientist level to really discuss what is our strategy, what assays and methodologies are we using, what are their limitations, and have open discussions on that, and basically really be perceived as not as someone that stops a project or comes with the bad news, but rather as someone that provides survival of the project on the long term. And I think if you can be perceived as such, you'll be respected and accepted as an equal team member, and medicinal chemists are making efforts then to optimize on safety. I think otherwise it is of course important to. I mean, toxicology is traditionally very separated from the drug discovery organizations, to make sure that people are sitting close together, that a discovery safety or exploratory toxicology department has the competencies not only of toxicology and drug development going forward, but also understand the drug discovery process. I think those are the key things. There's more, but I think those are the key things that I could share quickly.
Okay, great, thank you. Josh, a question for you, and then I think we have one more to wrap things up, or one or two more to wrap things up. So, Josh, how can core be used to efficiently discover biomarkers?
So basically, similar to the study that Richard described. So if you have an endpoint you would like to be able to predict with a set of cheaper in vitro assays, you can basically run the endpoint through our system and then look at what data in the knowledge base is highly correlated with that endpoint, and then should use your expertise to look at the specific assays and determine if there is some biological validity to those correlations, and then you can determine from there whether those should be used for in vitro assays.
Okay, well, thank you very much. Richard, you're the second to last question here. how did you identify negative compounds for your training and validation sets, since a study report will usually note the presence of a lesion, but not usually the absence of one?
That's a good question and a really important point in that, most publicly available data on safety is positive, and it's really hard to find negative compounds where somebody's exposed an animal to a compound and looked for a lesion and then not found it. That's typically not reported. In the ToxRef DB database that EPA put together, it comes from a lot of quite rigorous study reports. So you will know from that study report whether or not a particular set of evaluations was done, and that will include, whether histopathology was done in a particular organ. So you have to combine that together with the observational data and say, well, for this compound, they did look at histopathology and they did look in the liver, but they didn't see necrosis. And although it's not, 100% certain, you can assume with some quite high confidence that compound is therefore negative, because they looked for it and if they'd have seen it, they'd have reported it. So that's about as good as you can get. And the ToxRef DB database is quite a good resource for finding that type of negative information, which is otherwise quite difficult to find.
Great, thanks so much. So I think we're at the last question, unless someone else has anything else additional to add. And I just have one more thing to do after the last question, and that is to tell you about the opportunity to participate with Quantitative Medicine in doing a project for the Technology Evaluation Consortium. So hang on, and I'll tell you all about that in just after this last question, which I'm going to direct to Josh. So can the core aggregate data across the phases of a single research campaign, as well as across multiple research campaigns, or can core aggregate data to enable early detection of negative compound characteristics? If so, I would think core can sharply reduce later stage attrition. Can you comment on these observations?
I think that's absolutely accurate. One of the goals of building the knowledge base was to incorporate all kinds of information from many different phases so we can do exactly that sort of work. And so yeah, I think it's absolutely the case.
Great. Well, thanks very much to our panelists today. And I'm just going to recap by saying, again, at 1 Million Solutions in Health, we're here to help shape healthcare by sharing solutions. And just to remind you that there is an opportunity for organizations to participate in the Technology Evaluations Consortium on one project. Right now, today, you've heard about our project with Quantitative Medicine. If you attended our webinar last week, or if you'd like to know more about it, we also have another project you could participate in, which is an in vitro tool to evaluate the efficacy of cancer medications with Vivo Biosciences. So either of those are available right now for you to try out membership. If you want to just jump ahead and participate in both projects, we also have 2 other projects underway right now, and that would require you to join us for full membership, and you can participate in all 4 projects. The 2 additional projects, so if you participated in the Quantitative Medicine project or the Vivo Biosciences, because I just got a question, that would be for free. If you want a full membership, you'd have to pay for full membership, and then you would get those 2 projects plus these other 2 projects listed here. One on an in vivo liver toxicity assay that we're evaluating, and the other one, a nanotechnology to turn insoluble compounds into soluble compounds. So in order to participate, you would just need to contact myself or our Executive Vice President, Brett Johnson. I don't have his email up there on the screen, but both of us are the people to help bring you into the consortium. So one last question to see if anyone has any additional questions. Also to remind you to fill out the survey that's in our box, that's in the chat box. And I'm just going to go back to here, the panelists, to just say thank you very much to all of our speakers today. Josh Kangas from Quantitative Medicine, Jort Hornberg from Lundbeck, and Richard Brennan from Sanofi. It's been a very informative session, and I think lots of people will be interested in hearing this afterwards. And we do always record it, so we'll make it available to everyone. So any last words from the panelists from today?
Just want to say thank you for the opportunity to give this presentation, and thank you, Joel and Richard, for all your help and the excellent collaboration.
Yeah, thanks, Dawn, for putting this together and for the invitation.
Also, thanks from Jorrit here.
Great, thanks guys. You did a terrific job, and we're looking forward to hearing more exciting utilization of quantitative medicines technology. Thanks everyone for sticking it out to the end, and again, let us know if you'd like a copy of this. We'll be sending it out to everyone who registered, and it's available for view on our website shortly. Have a great day. Take care now. Bye.